We really appreciate having Hongzhou with us. As many of you realize or saw in the news, I think we actually knew it before he did. Lilly just did a reorganization yesterday, and unfortunately about almost 6,000 people. Well, they said 5,500. Will be made redundant. And as he's saying, he didn't know until he got here and called home. So I think we have him with us, and hopefully we'll have him yet for a while bit.
Yeah, I still have my badge, so at least I can go back to my office, right? So.
Well, he hasn't been back to find out whether it works or not.
All right. So, all right. Good afternoon, everybody. So basically today I just want to talk about some internal development effort we have. So we developed an application called SPrime, and we try to use it as our internal computational chemistry framework for drug discovery. And in this application, Marvin actually is one of our key components to improve the usability and also to extend the functionalities. So here is basic outline of this application, this presentation. And first of all, very briefly, problem and challenge. And then we move to the system design, technology integration. And then we just talk a little bit detail about different components in our application. And finally, we just use an example to put all the components together, followed by the summaries. So yeah, everybody know, pharma industry faces tremendous pressure, and from government, from general public to reduce the cost, provide better medicines, and also from Wall Street. So we failed this time. So in that case, in-silico technology actually was try to resolve, at least from a drug discovery process, to reduce this kind of cost. But at least at this point, existing in-house in-silico solution is really not up to par. And they are really scattered around, and they have a different interface, and they don't have a communication between different tools. And in our 2007 service, we actually have, just in the discovery chemistry department, we have over 100 tools. And some is GUI-based, some is web-based, or command line-based. A lot of these tools really just created or developed by the computational chemists who actually support some specific project. So because this kind of the scatter around, and the maintenance cost is quite big, and also it's hard to change the data. Most of the times, user basically use a file, load the file, and run the tool, and export to a file, and export to a different tool, and run again. So it is quite difficult. So learning curve is big. So for the chemists, really, they don't have too much time to do this kind of things. And if this thing is hard to use, and they will not use. So in that case, all those kind of routine work will fall back to the computational chemist. And therefore, they don't have enough time to do their high-value project support work. All right, so we have some proposed solution to basically resolve this kind of the problem. And first of all, we try to provide a robust framework to support in-silico technology. For example, if you have a new models, you have a new tools, and at least it's much easier to add this kind of information into the framework. And also, we want to have a single data representation, such as, different tool, different model, they can exchange the data. So users don't have to go in, go out, go in, go out, right? And also, we want to provide a single integrated interface to access all those kind of the computational science, to do data analysis and to do some visualization. Basically a single interface and rich interaction. And because it's kind of a single interface, we actually can reduce learning curve. So chemists, hopefully, when they pick up one tool, and they can actually pick up another tool. So increase the usage of the tools. Finally, basically, you basically provide this kind of environment to allow the user to basically create some idea and validate the hypothesis and to try the drug compound. And basically, they can do this kind of virtual experiment before they move into the web lab. And finally, basically, with this application, we can retire or replace some of the internal application to reduce the cost. So here is really just quickly logical design, and it is a classic three-tier system. We have a client which deploy into the user's desktop, and we have a backend on the left which sits in the most likely in the Unix box, and some service was on the Windows box. So all those kind of the server was wrapped in the web service, and some web service take advantage of the cluster to do some of the computational intensive work. And you can say, okay, between the client and the middle tier, between client and server, they are really just a many-to-many relationship. So this middle tier in the middle actually bridge client and the server. So the middle tier really just got the request from the different client user, and based on the request, it can just send to the different server. So when the data returned, it is this middle tier to actually convert data to our internal data representation and associate author to this data and save in the database. So client, basically user, they can fire up and go. They don't have to wait until the result is finished. So to do that, to increase the data exchange, and we actually define the unified data source as our SPrime internal data representation. And basically, some rules, all structure got standardized when they enter into the system, and all the import structure was converted, or import the data or structure were converted to this we call UDS, and all the backend return data was also converted to the UDS. So this UDS really just a single data representation, and we actually bound this UDS to a data grid for the data visualization. And also we have a standardized plugin mechanism to add new feature. So therefore, it's relatively easy compared to before. If you have a new type of model you want to add into it, if you have a new type of the Cheminformatics tool you want to add it, it's much easier. So for developer, they can just spend maybe a couple hours and they can just finish this part. And finally, always connect the mechanism. So I think this one is a very convenient. For example, you set up some meeting, talk to your project team, and before you go, you actually run some job and unplug your computer and run into the meeting room. When you get there and connect to the internet, and all the job were there. So basically, it's encourage the people to do more collaboration. So this slide really just itemize all kind of the component in the application, and some of the component will be discussed in the later slide. So I just don't want to spend much time on these things. So one thing I want to emphasize is our just the job manager, because for now we can support up to 40,000 molecules, and for this kind of the big data set, our job manager will split large compound set to a small different task, and we can run locally or we can run in the cluster, and we can combine the data later on. So it provides some the freedom for us to do this kind of work. Quickly go through technology. And it is a hybrid environment. So client side, C#, C++, Java code, and the server side, because most of the server was in the Unix, so the Java is dominant, but also we use the Perl or use the SAS R to do the model building and model prediction. And also we integrate all kind of the commercial or open source components. Commercial one like Marvin or DevExpress data grid, and we use GenBridge, and also use semicontroll object library to manipulate some structure, draw structure. And open source like ZGraph to do the charting, and JMO for the 3D-based data visualization, and of course, Tomcat web server to host our web service. Some internal component as well. So we have a huge amount of the C++ library to do all kinds of the Cheminformatics. And they're actually running in the Unix for a very long time. It's very robust, very speed-wise, very good. So we actually import some of those kind of the tools into the Windows side user mainway to take advantage of the desktop the performance. And some function like structure standardization and substructure search and fragmentation reaction. So here basically how we actually use Marvin in our application. So Marvin actually was very good, which provides intuitive structure joining functionality. And also in the 5 version, you can do some customize the UI to simplify the joining process. But at least 2 years ago, when we started this project, it's Java-based, and our corporate requirement really to use C#.NET for the GUI-based application. So cannot fight over. So basically, we search a different solution. And at that point, we found, okay, GenBridge is actually the only technology which can actually embed those Java swing object into the C# control. But in the early version, we have spent quite an effort to deal with this kind of frequent crash in the early version. And many times when you click a more button in Marvin, it's basically the freeze or crashed. And also in the GenBridge, it makes last event. So many times when you try to resize, it didn't go through it. And by working with a developer in the first company, and we actually found some workaround, and we add some internal timer to add that additional event, and also we upgrade Marvin to 5.1.7, and because our corporate GRE environment is 1.4.2, and we cannot go higher because of that kind of limitation. And also we use the Marvin to generate a structure which serve as a tooltip. So whenever user move around their mouse, and that structure will generate on the fly. And actually, that one is quite good. And also, Marvin Sketch was used as a part of tool interface. So we have several tools like Structure Examiner, which will be discussed later. We have a Reverse Cursor or Property Explorer tools, which all use Marvin Sketch as a part of interface. So finally, Marvin plugin actually supplies several models like log-P, log-D, and PK models. So before I move to some of the individual components, I just want to emphasize 3 basic entities in our application. So we have a compound set, we have a model, and we have a tools. So basically, model 2 will act against the compound set to generate another compound set. So this compound set really can be recycled, and this compound set was actually converted to the unified data set, which serve as an internal data representation. All right, so here is one component called Predictive Models. So at this point, we have about 500 models available in SPrime, and it's displayed in the tree format. We have a global model, local model, and also you can organize based on type. For example, we have some even near neighbor as a model, and we have a 3D docking model, functional model, or even just this regular in-silico models. And basically also group as a global model, which actually available to everybody but cannot be modified, or local model, so that's really belong to a single user, so they can do whatever they want. And because most of the project really just involve a lot of many models, so we actually create this model folder. It's kind of like a file folder to group model together. So basically, the computational chemists really can just play around with your collection of models. And at that point, all the model is private, so nobody else can see it. And once they think it's ready to be used by the public, by the team member, they can just make it public, or they can just publish their model group, which are associated with a specific project. So in that case, chemists in the project, they can really just find out the project and check all, so everything was ready, instead of just every time you just select specific models. And we also do some post-processing and the consensus model. Easy way to understand the post-processing, just as an example, like thermodynamic solubility model. So most likely the data was log S, right? It's in the log unit. But chemists are more familiar with micromole or microgram per mole. So you need to do some of the transformation after your model prediction is finished. So we have this kind of the formula engine. Just the interface was basically with here. So you can actually define how you want to transform your data. And another one is a consensus model. If you use a multiple model, like when you deal with metabolism, so we have a human model, we have a mouse, we have a rat, we have a monkey. So sometimes you want to actually get some consensus between different models. So this engine can be used to define that kind of consensus. And when you run this model, it automatically generates a consensus to you. So another use really for the consensus model is for the score function, right? So it's kind of like multiple parameter optimization. You can assign a different weight to the different model, and in the end, you have a single value to follow up when you try to optimize your compound, whether in the property space or activity space. So no application really can do everything. So you have to rely on the third-party application. So you have to provide import, export functionality. So we actually cover relatively good, and you can import structure from Lilly IDE or MDDR IDE or Smart SD file. And you can draw structure, use SymCdraw or Marvin. The reason for SymCdraw is most chemists really familiar with IC-Draw, so we put over there. But personally, we think Marvin's sketch is better. And also you can enumerate the structure either through the fragment or reaction. And also you can generate fragments through some internal bioassay tool. And finally, you can actually import some 3D data, which can be served as an input to our 3D tools. Export function, you can go to text file, Excel, spot file, Smart SD file, so that's basically standard. Because for now we have all the source code, so if our business partner needs some special format, and we can actually do it for them. For example, we can export the data as a star format, and they can actually load this data and run some star job for some other analysis. So when you select a model or you select two, you run the job and you got the data. So the data was in the format of unified data source. And this data was, we actually use a DevExpress actual grade to display this data. And we support number, text, and image. Like, for example, you generate log D image, we can do that. And we have a special type of model called wisdom, and you can show the near neighbor of fragment and a 3D structure. And you can do some sorting, filtering, and substructure search. And also we provide a chart. So you can basically right-click grid, you generate a chart, XY chart with a regression line, and you can create some text chart and bar chart, SAR chart. So this chart was developed on the top of the Z graph, which is basically the open set. So the good thing here is really between the chart and the data grid, they are actually synced. So whatever you do on the data grid, it will be reflected on the chart. Whatever you do on the chart will be reflected on the data grid. And always, when you move your mouse over, the structure will be shown. So as a chemist, you know your structure, and you can see the whatever response you do when you actually do whatever, how to say that. So some advanced data representation. Like I said, we have a special type of model called wisdom. And in that models, you can actually show the training set. For example, like here, it's really just a classification model. So we can define this zone as an indeterminate zone. Basically, for the modeler actually can inject some expert call into the model prediction. If your model actually falls into this region, this green region, that basically means, okay, your prediction is really not reliable. So we basically give some model some leeway to do that. And here we can see, okay, which part of fragment actually are not covered by the training set, and which part of the fragment which actually make the most contribution. So chemists really can say, okay, all right, this part of the fragment really is not useful. I can actually just cut it out and just add some other things and to see how it works, right? And finally, we can show some basically near neighbor for every compound when we make the prediction. So user can get another feeling to say, okay, how good of your prediction is. Many times, okay, when you got the data, the user really want to say, okay, I want to do some annotation. I want to add some comment on the structure. So we allow the user to add a new column, either text, so they can just type in, or number, or they can just use the formula I displayed previously. They can just do some calculation on the real time, do some coloring. And because the data was unstable at that point, so we actually have a two mode. One is edit mode, one is a normal mode. So some functionality is not available in the edit mode, so you have to get out before you do that. So we have three entities: compound set, we have a model, and here is the tool. So we have a lot of the internal tool parties or just the developer inside this SPrime, like design tool, have some structure examiner. It's kind of like structure analysis or SER analyzer. Dicer, we call dicer, but it's kind of like a fragmentation. And a TI action, like your reactor, but it's just an internal C++ code. We can do some scaffold-based clustering. And some 3D tools, like when user loads some compound set, they can just run some docking model or run some function model. So when the job was finished, and they can just bring data in, and they can do some analysis, use the Gmod here. And also, they can just use a video to do some of the analysis. Property Explorer, which actually uses a Marvin sketch as a part of interface, is really to allow chemists to iteratively draw some compound and make the prediction, and then draw another compound, make a prediction, and basically kind of like immediate feedback mechanism, so they can just gradually to optimize their structure. All right, so that's basically the component I want to talk about. And here I just want to use one example and to put everything all together. So we use a Marvin to create this customized library enumerator, and that's done maybe in 2 or 3 years ago. So user can really just draw the call structure, and they can draw the R group, whether the R group come from their experience, come from the literature of patents, or even come from the bioassociator tool. So they can draw those things, and then they can just, when you click, you just separate into different panels. So R1, R2, R3, call structure. At this point, user really can just say, okay, I want to maybe remove one of them, add another additional R group, so whatever they can do. And after that, you enumerate, and you got this kind of nice, great looking library. So it is really just a snapshot of your libraries, and you can show, okay, which R group, which call group you use, and here you can, depend on how many R group you can have, you can basically to switch from R1 maybe to R3, and structure got basically updated accordingly. So user really can just play around and to say, okay, the snapshot of your library, and when you're done, and you can just add libraries, it will be added into this interface. So at this point, it's really just a regular compound set. And once you got this compound set, and you can actually select some models. For example, if you are in the lead generation stage and you have some hypothesis, for example, like your activity really positively correlated or negatively correlated with the log P, you can select that models, several models, and make some predictions or run some tools. So after you've done that, you can create some XY chart and to see whether your activity really correlates with your log P. And this one is we call the text chart. So this one really shows you, say, for the R1 group, I have many R1 group, which one actually makes the most contribution, which one is not. So you can see that immediately from this chart. And this one we call SY chart, called SAR chart. So that's really just to dump the whole library into the two-dimensional space. And it's kind of colored based on the property you selected. So at this point, you can see immediately, if you like, okay, the lower C log P, the better, and you can immediately see some of the hotspot, and you can actually drill into it to see what kind of structure you have. So move your mouse around, and you can see the structure. So like I said, okay, the data grid and chart linked to each other, are synced. So you can actually do some of the data analysis or manipulation in the data grid, and your chart will reflect your operation. You can group data, and you can sort the data either by the single column or multiple columns, and you can create some of the customized filtering stuff, and you can do the substructure search. Here we have a, where's my mouse? So here we basically convert to the smarts for your query compound, and you can switch to the different things, and you can create some subsets, or you can combine the data all together. So basically, in the end, you can generate the preferred subset of your compound from these libraries. And once you get that libraries, a subset, you can go back to a tool, right? So you can run the docking tool if you already have a docking model available for your project, and you can run functional models and to basically get a better idea. So hopefully, okay, with this tool, because all those kind of functionality you can do it in a single tool instead of just switch over again. So hopefully chemists, once they learn how to use these things, and they basically can pick it up quickly. Hopefully they can spend more time on this kind of in-silico technology before they switch to their wet lab. Finally, okay, summary. So we deliver this desktop-based solution to allow scientists to access in-silico technology to generate hypotheses, and hopefully they can make informed decision earlier. And it is a single click to start application, so they don't have to. It's basically was you can it's an icon on the desktop, and also it's available start menu, so they don't have to remember where to find this application. Automatically update, and also it's very easy to report problem or get feedback. So just tap in and click button, and we receive it. And also I think the key message is really just a quick response to the customer feedback. So most likely, and we actually respond in 1 day. And something is easy to remember or easy to resolve, and we basically, 1 or 2 weeks, we can just patch that part. And we actually officially released to the chemist community in the last year, August, I think. So basically, within 6 months, we got over 100 users. Now I think it's close to the 400 users, and if you calculate, it's about 500 penetration to the chemist communities, and normally, so every month it's about 200 more different users run the tool, and they can run over maybe 1,000 or up to 2,000 jobs every month. And we also retire several in-house applications already. So finally, acknowledgement. The folks at this side really just the part of the development team, and Ian Watson really just developed most of the C++ library for the Cheminformatics tools. And this project also was collaborated with a company called HiSoft in China. So these folks really focus on the middle tier and the client-side development, and people in ICS convert one of the wisdom model into this application. Dr. Johnson, Dana, a chemist who actually run the training class, and people here is really just to provide some feedback. And yeah, okay. So that's basically the last page. And thank you very much for your attention.
Thank you. Are there any questions?
Take about half an hour.
Just a moment. Yeah. Pat, I think you can start without the mic.
Okay, yeah. So when you look at the typical, I mean, it seems like a lot of what you showed there was regression models.
Yeah.
And when you look at the typical dynamic range of bioassays and the fact that you can have 3-fold, 5-fold error,
yeah,
it's really hard to do regression.
Yeah.
So
yeah,
I'm wondering, sort of,
yeah,
you said you've got 500 models there.
Yeah.
And what fraction of those are regression versus just a classification model?
So a lot of it really just a simple model. For example, you lipid schedule of 5 and log P, log D, those kind of things. And although we have maybe several dozen element models, and we have an activity model based on a different platform, but most models really are local models, because like you said, the data itself, the data quality is a big problem, right? Yeah. So a lot of assays really didn't didn't allow you to build a global models. So most likely people will say, okay, let's find some data for this specific project and build the model for that specific project and use that. A lot of times, really, okay, the model really didn't perform good. So it is up to the project team say, okay, this model, at least it didn't work for me, didn't work for this project. And some model maybe work for another project and they will continue using it. If not, they just didn't use it. So that's reality, really.
Right. So I'm sorry, I didn't really catch this at the end. So what fraction of the chemists at Lilly are using this right now?
Okay, so that 360 users is, I think it's 2 months ago. I think at this point it's close to 400.
But what fraction? I mean, is this half of the total or
yeah,
a third?
It's about 50%.
Okay.
Yeah. So I think not bad. So we released last August for a year. So
that's great.
We actually pick up quite quickly. Yeah. So.
That's a tremendous adoption. Yeah. Once again, I want to thank you for your speech. Thank you very, very much.